Hi there, this is Mr. Waite, and I wanted to go over with you Unit 2 Cell Structure and Function Study Guide real quick because there's a chance I may not be at school tomorrow, October 1st. So if you would, make sure you have this out in front of you, and let's highlight a few important things. And then, as always, if you have any questions, you can either email me or come see me before the test. Either one is great. So let's get started with this really quickly. If you have your study guide, I'll highlight just a few things. We'll start out with ribosomes. Please make sure you remember that ribosomes are found in all forms of life. They're in prokes, they're in eucs. We've got the ER. Rough ER is where the proteins are. Smooth ER, they do detoxification and lipid synthesis. Golgi complex, I'd like to think of as kind of the UPS center of the cell. They're going to package the proteins up, send them out to the cell. Mitochondria, this is important. They have a double membrane. Maybe I could draw it on here if I had two membranes, the inner and the outer. Lysosomes, they have those hydrolytic enzymes that go around cleaning things, hydrolytic enzymes. And then vacuole. Last thing is chloroplast, found in plant cells or algae, and they should have it down here. Here it is, another double membrane. Double membranes found in both those, and those are important because we're going to come back to those here in just a little bit, especially at the end when we talk about the endosymbiotic theory. So let's keep looking at some other stuff. 2.2, their structure and function. ER, I feel like we've just talked about. Mitochondria, double membrane. Again, some of this is pretty repetitive. And I think this is the last thing down here that's super important, okay? The folding of the inner membrane increases the surface area. This is what it's all about. Let's move this so it's out of the way. I apologize. So these folds increase the surface area. And when you increase the surface area, you're increasing the amount of reactions that can occur. And that's super important if you are making energy, if you are producing sugars. So keep that in mind. Next thing, some pictures. Good. So we've got the prokaryotes up top. Just good to recognize these are the prokes. Down here is a eukaryotic cell. And with the prokes, what you'll see is we have DNA in one part of the cell. May not be a nucleus, but it's at least localized there. And the only other thing you'll really see are the ribosomes. So we've got DNA, ribosomes, and a cell wall. But down in the eucs, we do have a very specific place down here for our DNA. That's in the nucleus. You will not find a nucleus up in the prokes. In addition to the nucleus, you'll find other membrane-bound organelles like the ER, rough ER, the Golgi apparatus, a lot of other things, because if you are wanting to be extremely efficient, you've got to have each thing compartmentalized, and you want to have lots of room for area, you've got to fold those as well. So you increase the surface area, increase the amount of reactions that can take place. All right, last thing, well, not last thing, but one of the other things that's on here is the cell size. Let's find the important thing on here. Large enough, adequate. I'm reading it through here with you. There it is. High organism have used specialized cells. I wanted to find, here it is, as cells increase in volume, the one above it. Smaller cells, so I'll start right here, have a higher to surface volume ratio. Okay, so it's good to be small. Now you can see down here in this picture, I can't draw on it, but the small cell that has a surface area of 2 centimeters, the SAV ratio down here is 3 versus the large cell over at the end with 8. You can see it over here. It has an SAV or SAV of 0.75. So the larger the ratio, the better for the cell. And how do you get that? By being small. So it's almost it is an inverse proportional relationship. Then we're talking about the plasma membrane, and don't forget it's fluid. I'm looking for things in here. Most important, it has a polar area and a non-polar area. I can't draw on the picture, so let me come off the side over here. So if I could draw this membrane right here, I like to put NP in the middle of it. Okay, so we know we have a membrane on top, the membrane down here. NP is non-polar. It's like the no-fly zone. You can't get through here. That would mean up here it's polar, and down here, down below, it's also polar. So hydrophilic, water-loving, but in the middle down here, we've got our hydrophobic, almost water-hating. Don't forget things like steroids are super important. They're going to give the cell structure. They're going to keep it together when it gets cold, when it gets hot. But those are all good things to remember. Hydrophobic, hydrophilic, and again, polar can just mean something as simple as being charged. Next stage is membrane permeability. What's going to get through our membrane? Don't forget it's selectively permeable. It doesn't decide. It gets to have say over what goes through and what doesn't. And it talks about things that can easily pass through. Non-polar, non-charged, small. These things, this is important because things like oxygen, things like carbon dioxide, they have to be able to get into the cell, and carbon dioxide has to be able to get out of the cell. So general rule in effect is you're not going to get through if you're large and you're charged. So that would be this purple arrow down here at the bottom. It would turn around, versus if you are small and non-polar, like the green arrow right here, you're going to pass through that lipid bilayer pretty easily. Some things, if you can see this third arrow that's blue, water sometimes will get through in small amounts, but it won't get through in large amounts without the aquaporin, and I believe we're going to talk about that here in just a little bit as well. So let's get rid of this. And then we have passive transport. Passive transport, import materials and export of waste. This is good to remember. High to low, high concentration to low concentration. High to low is the way to go. Again, high to low, way to go. Other things up here, active transport. When I talk about active transport, we really need to know we're talking about you're going to require ATP. And that's, again, for active transport, because most likely you're going to be going from a low concentration to a high concentration. When it does go from high to low, we refer to that as the concentration gradient. And again, if you want to go against the concentration gradient, you're going to need active transport and ATP. Two things that are important to memorize to remember through this process are endocytosis and exocytosis, taking large stuff outside of the cell. Exo means to exit, endo means to come in. Next thing we're looking at is facilitated diffusion. You can see this down here. Facilitated means you need some type of a helper. Usually that's in a form of a transport protein or a channel like this. The other thing, remember we talked about water cannot pass through. Small bits of water can sometimes diffuse through, but if you're large quantities of water, you're going to need these things called aquaporins, a type of protein channel that's going to let large amounts of water through. Other stuff on here, I'm looking, metabolic energy is required for active transport. And then they talk a lot about the sodium-potassium pump, but we really didn't look a whole lot at that. Let's keep looking for 2.8, tonicity and osmoregulation. Super important words on here. Hypertonic, hypotonic, and isotonic. So if we can identify those really quickly, it's always referring to the environment that a cell is in. And unfortunately, I can't draw on these pictures down here, but if it's hyper, there's more party on the outside. Let's just do a quick drawing of what that might look like. So if this is my cell in solution, let's imagine the solution is like this. Lots of stuff on the outside, very few things on the inside. This would be hypertonic. Now, opposite of that, if I came down here and I drew a few things, I say there's lots of stuff inside of the cell. We always refer to the solution around it. This would be an example of a hypotonic solution. Hypo means low. There's not very many things around here. And then, as always, what's important is which way is the water going to go. Water always go where the party's at. It's going to go outside here. It's going to go inside here if it was in a hypotonic situation. The exception to both of those would be if it's isotonic. where you have equal amounts or concentrations of solutes on the inside of the cell and on the outside of the cell as well. Osmoregulation, controlling the internal solutes. Don't forget we talked about that in fish, saltwater versus freshwater fish, how they maintain their internal water environment. And also don't forget, I stole a picture of this over here. This is from the formula and equation sheets. We don't want to focus too much on this. If you had to do a problem or two, you should at least be familiar with it. On the test, you will be given those, so it's not something you have to memorize. I'll give you a sheet with those equations on it, and if you have a question, don't be afraid to ask. Let's keep looking at these. Oh, right before I leave this, don't forget animal cell in a hypotonic situation like distilled water would become lysed and explode. Here's isotonic. In a hypertonic solution, all the water would escape from the cell and go into the solution. This would shrivel and shrink. Animal cells are different than plant cells because plant cells have this rigid cell wall around them. You don't see them shrink necessarily, but you could see the cell membrane pull away from the cell wall. We call this, they have the word down here, plasmolysed. Okay, kind of interesting. Flaccid is normal, and then turgid is when you have lots of water inside of there. You're not exploding necessarily because you got the rigid cell wall, but it still can hold large amounts of water. We talked about moving things across. We already talked about endocytosis, exocytosis. Those are both forms of active transport, which requires ATP. 2.10 compartmentalization. Again, it's all about making the cell efficient. The more enzymatic reactions that you can have, the better. You're going to be more efficient. And how do you do that? By having more surface area. How do you do that? By having lots of folds. That's especially evident in the chloroplast of plants and the mitochondria of both plants and animals. All right, wrapping things up here. 2.9, 2.10, the last one, 2.11, when we talk about the origins of these cells. Mostly we're talking about mitochondria and chloroplast. Where do these come from? We have the theory here, good to know. Tests love to ask you about the theory of endosymbiosis. And don't forget to break it down. Endo into sim, together, bio, living. Something goes into something, it doesn't become digested, and they learn to live together. Sometimes I've asked students for a definition of endosymbiosis, and it's not simply that one cell swallows another one. You have to talk about how not only does it get swallowed, but then it also continues to live in harmony with the other one symbiotically, and helping it achieve a greater purpose. For example, down here, this is our idea. We think an early cell engulfed early prokaryote that eventually could produce energy. This would become mitochondria. Same thing if this were a plant cell, engulfed another one that would make food. This one would be the chloroplast, and we also have evidence for those. Some of those evidence were that mitochondria and chloroplast both have their own prokaryotic ribosomes. They each have their own circular or prokaryotic DNA, and most importantly, don't forget, they have a double membrane. How would you get that double membrane? If you have your own membrane and then you get engulfed by something else, you incorporate that membrane as well. Guys, if you have any questions, be sure to let me know, and otherwise, good luck. And don't forget, bills are due tomorrow or the day of the test as well. Good luck. You can get this.